Assalamu alaikum students. Before going to our educational topic, please subscribe my channel Muntzir Mehdi Science TV on YouTube. Press the like button and share this lecture to your friends and classmates who have no access to it to add in their science education. And those students that are preparing privately can benefit from our channel very effectively. Let's come back to our topic, that is enzymes action. So in this tutorial video we will conversation about mechanism of enzymes action, namely by which methods does an enzyme work. Dear students, basically two models are described in our FSC and BSc levels, that are lock and key model and induced fit model. In this tutorial video we shall understand both these models ably. To understand the mechanism of enzymes action, first of all, this is our enzymes diagram, that is represented by E. An enzyme requires a thing, a substance, to perform its catalytic activity, that is a substrate. This substrate molecule comes to be attached to the enzyme. The substrate molecule is represented by a letter S. Students, to understand the enzyme's action, the presence of substrate molecule is essential. As I have already described in my previous video lecture, that anything upon which an enzyme will act is a substrate for the enzyme. For example, if an enzyme will act upon a protein like meat, beef, or chicken, then protein will be the substrate for the enzyme. But if an enzyme acts upon the carbohydrates like wheat's breeding, then carbohydrate will be the substrate for the enzyme, and so on. Therefore, that particular substance upon which an enzyme will perform its function, its job, is a substrate. So then when enzyme and substrate attach, students, generally I am talking about the attachment of enzyme and substrate irrespective of their specificity, that which type of this enzyme is and which type of this substrate is. Either it is a sucrase or lactase enzyme and pepsin or trypsin enzyme. Either it is a carbohydrate or protein substrate. For general understanding, I am saying that the enzyme will bind to the substrate or substrate will attach to the enzyme in this form, that we will get an enzyme substrate complex. Students, substrate is bound at the specific site of the enzyme, that is known as enzyme's active site, and is present only here at this point. Although an enzyme has a large surface area, but whole of the enzyme is not functional. Only a tiny portion on the enzyme surface is active, which provides a site for the attachment of a substrate, that is known by the name of the active site. So after the achieving of this ES complex, that is the short name of enzyme substrate complex, the enzyme will start to perform its catalytic activity. Let's suppose it will start its breakdown, its splitting. Students, listen. At the FSC level, most of the students consider that the enzymes are only responsible for the splitting of things, splitting of different compounds. But it is not the reality. Enzymes are also involved in the attachment of things, in the binding of compounds, different materials. So they can also synthesize the different substances, different compounds. So here for the substrate, we have assumed that the substrate is splitting and is changed into these smaller fragments. Now these tiny fragments upon which the enzyme has performed its function are called product. So now it is our product, and the enzyme will free as it was before. It will not being used up during the enzyme's catalytic activity. Now this free enzyme is ready to be attached to another substrate so that it can catalyze another chemical reaction. Students, as I have already tried to understand you that with the enzyme doesn't happen, it is not affected during the catalytic activity but only move forward a chemical reaction. As you can see that here the enzyme is the same as it was before, because if it carry any change then I must describe it. Students, next we conversation about two models of enzymes. Students, there are two models of enzymes that were proposed by two different scientists. The first model is named as lock and key model that was prepared in 1890 by Emil Fischer named scientist. As the first enzymes was discovered during 1833 year around, that was diastase by working upon starch, that is a carbohydrate, and converts starch into maltose. This first enzyme diastase was discovered by a pine named scientist. At that time, diastase was not known by the name of enzyme because later in 1877, W. Kohne gave it the name of enzyme. This word enzyme is made up of two words. In means inside and zyme means yeast, that is a fungi. Students, as the first enzyme was discovered in fungi, so due to this similarity, it was named as enzyme. It is a bit detail about enzyme. The purpose of telling this detail is that after a large time period gap, the first model of enzyme was proposed, whereas after many years, about 68 years later, second model of enzyme was proposed by Kochland in 1959. So we can see that a long time period is required to achieve a thing and by proving it. Firstly, we see the lock and key model. Dear students, in the lock and key model, it was compared by a lock and its key. Because according to this model, the active site of an enzyme is a hard, rigid structure. Because nothing change can happen in the rigid structure of the enzyme's active site. That is the first point, which shows the enzyme's specificity. Students, specificity means that an enzyme can catalyze a specific chemical reaction. It can change only a specific substrate into a product. Students, if you want to understand the specificity of an enzyme in more detail, then you must watch the characteristics of enzymes video, that is the previous video, during which I have described this term in more detail. Students, to understand this concept well, consider that as a specific key can unlock only a specific lock. In the same way, a specific enzyme can catalyze only a specific substrate during a specific chemical reaction. Because according to this model, an enzyme can be compared with a lock and a substrate can be compared with a key. So due to this comparison, we can say that like a specific lock's own key is required to unlock this specific lock, because this lock's own key that a specific lock possesses, whose figure frame is specific to fit into the socket of that specific lock, can unlock that particular lock. Likewise, a specific enzyme can catalyze a specific substrate, bind with a specific substrate. Other substrates can't attach with it. In this way, this model is describing the specificity of an enzyme on one hand, and on the other hand by the lock's comparison, which has a rigid socket like the enzyme's active site, which can accept only lock's own key to be unlocked. Because if we use another key for unlocking that lock, it can't unlock it, because that other key will be the other lock's key. As every lock's socket is a rigid structure made up of hard material which has no elasticity. If it possesses a little elasticity, then by applying the force, we can unlock the lock. But due to its rigidity, every lock unlocks by its own key. This similarity is adopted to describe the lock and key model. Secondly, I will describe the induced fit model. Students, induced fit model tells us that the active site of an enzyme is not a rigid, not hard structure, but is flexible, which possesses an elasticity like property. Suppose this is our enzyme, which is according to induced fit model has a flexible active site. As this induced fit model was proposed by Kochland on the basis of new evidences, so it has a flexible active site when a substrate of this shape comes to be attached to the active site of this enzyme, which is completely different from the substrate figure, then you can suppose that a different shaped substrate can't fit into a different shaped enzyme's active site. Dear students, this was a major issue for the Koshland also. To solve this problem, he tells us that the enzyme remains inactive unless the substrate comes to fit into its enzyme's active site. So, before the attachment of substrate into the enzyme's active site, the enzyme will remain inactive, unfunctional. But when substrate comes near to the enzyme, then it induces a change in the enzyme's active site. So, due to this induced change, the enzyme will arrange its active site again in this way that the substrate molecule will fit into the active site of the enzyme. So now you can yourself watch that here the enzyme's active site is different, but here enzyme induces a change in the active site and allows the substrate to be fit into the enzyme and starts its catalytic activity. So students, till here we can conclude that the basic difference between the both models is of active site. Lock and key model says that the active site is a quite rigid structure that will not be the changed before and after the attachment of a substrate molecule, that will remain the same. On the other hand, induced fit model tells us that the enzyme's active site shape is different before the binding of a substrate, but after the attachment of a substrate, the active site becomes different due to a change that is induced in the enzyme's active site. Dear students, this induced fit model was compared by the glove in hand term in the same way as lock and key model was compared with a lock and its key. Here the glove is representing the enzyme and the hand is representing the substrate. Students, it is a very important point to understand the induced fit model. So put your attention to this whiteboard. Mostly gloves are whippiness, namely flexible, possess elasticity. So a glove can wear on the hands of different humans because due to its elasticity that a glove possess short, medium and large sized hands can pass into it and fit into it, providing that the glove must flexible. Therefore, they compare it with glove in hand term so that we can understand it wholeness. Students, next I will describe about the regulatory and non-regulatory enzymes that are associated to both lock and key model and induced fit model. To understand both these types of enzymes, we can say that those enzymes which follow the lock and key model are non-regulator, belong to non-regulatory category of enzymes, which means that those enzymes which follow this model, that is the lock and key model, have a problem that if they perform a large number of reactions are involved in the most of the substrates breakdown, then how we will stop them, that is a very specific and important point. They will not have any check and balance. Usually the enzymes present in our bodies possess a control, a check and balance. Students, usually when the enzymes are required to perform their functions, then they come at their specific function performing site where they perform their work. But when the requirement in the body is fulfilled, then they are inhibited from their jobs, from their functions. Therefore, the enzymes do not perform their jobs on their own, but they receive signals and they do their functions according to these signals. So, those enzymes which perform their functions according to the body control under the body's check and balance are known as regulatory enzymes. So, lock and key model represents that the enzymes which follow this model are non regulatory enzymes. So, there is no check and balance for them. An example of non regulatory enzyme is lipase. On the other hand, those enzymes which follow the induced fit model, the body can bring those enzymes under control under the boundary of check and balance. So these enzymes face a regular check and balance by the body functioning. Therefore, the enzymes which perform their jobs under the body's check and balance control are known as regulatory enzymes. The example of regulatory enzyme is hexokinase, that is utilized during the conversion of glucose to glucose 6 phosphate during glycolysis, which is also known by the name of glucokinase. Students, this enzyme starts its job by the delivery of signals. So by the receiving of signals, this enzyme starts their function and stops their jobs. So those enzymes which start and stop their functions by the receiving of signals, like the signals of deficient amount of ATP, are called regulatory enzymes. And students, regulatory enzymes work under the model of induced fit model. So students, it is our hodernal topic mechanism of enzymes action, which include two models. The first one is lock and key model and the second is induced fit model. As well as I have also described regulatory and non-regulatory enzymes. Students, finally I want to encourage you that do not waste your time that is very estimable and meritable to you. So engage yourself in your valuable education and other healthy activities. Lastly, many thanks for watching this your video.